Good day, this is Myla T. Sarmiento. In today's video, we will be talking about the normal flora of human body. Did you know there are more microscopic bugs living in your body than there are people on Earth? No, so, did you know that the ground we are standing on is made of living tissue? Yes, that's right. According to Michael Spector, we are inhabited by as many as 10,000 bacterial species. Those shall outnumber those which we consider our own by 10 to 1 and weight all told about 3 pounds, the same as our brain. Together, they are referred to as our microbiome, and they play such a crucial role in our lives that scientists have begun to reconsider what it means to be a human. What is the normal flora? Normal flora are the microorganisms that live in another living organism, such as human or animals, or inanimate object without causing disease. The human body is not sterile. We become colonized by bacteria from the moment we are born. We are covered with and contained within the intestine approximately 100 trillion bacteria that form the normal flora of our bodies. This normal flora helps to prevent us becoming colonized with more dangerous bacteria, which might lead to infections. The term normal microbial flora denotes the population of microorganisms that inhabited the skin and mucous membrane of healthy normal person from shortly after birth until death. It is the term used to describe the various organisms that are permanent residents of the certain body site. Their flourishing depend on the psychological factor of temperature, moisture, and presence of the certain nutrients. Specific of normal flora, these are include of bacteria, fungi, and also of viruses. There are advantages and disadvantages of normal flora. The first advantages is provide a first line of defending against microbial pathogens through bacterial interference. Interference may involve the competition for receptor and binding site on host cells, competition for nutrients. Assist in digestion. They produce vitamin B and vitamin K in intestine. Play a role in toxin degradation. The oral flora contribute to immunity by inducing low levels of circulating in secretory antibodies that may cross-react with pathogen. The microtuba affect the carcinogenesis in three broad ways. And these are the alternating the balance of tumor cell proliferation and death, regulating immune system function, and also influencing metabolism of host-produced factor, food and pharmaceuticals, such as the lactobacillus and phibidobacterial, are known to prevent tumor formulation. On the other hand, there are the disadvantages. They can cause disease in the following: when individuals become immunocompromised and when they change their usual anatomic location. Normal flora vary in both number and kind from one site to another, although the normal flora extensively populate many areas of body. The internal organs such as CNS, blood, liver, spleen, kidneys, and bladder are free of all but the occasional transient of organisms. The skin and mucous membrane always harbor a variety of microorganisms that can be arranged into two groups, and these are the resident flora and transient flora. The resident flora consists of relatively fixed types of microorganisms, regularly found in a given area at a given age. If disturbed, it promptly re-establishes itself. While on the other hand, the transient flora are the non-pathogenic or potentially pathogenic microorganisms that inhabit the skin and mucous membrane for a short period of time, like hours, days, and weeks. It is derived from the environment, does not produce diseases, and thus establish itself permanently on the surface. While going back to the tissue specifically of normal flora, the most membrane of the normal bacterial flora refers to colonize certain tissue and do not others. This tissue specificity is usually due to properties of both the host and the bacterium. Usually, the specific bacteria colonize specific tissue by one another of this mechanism, and these are the tissue tropism, specific adherence, and also the biofilm formation. Tissue tropism. Tissue tropism is the bacteria preference or predilection for certain tissue for growth. One explanation for tissue tropism is that the host provides essential nutrients and growth factor to the bacterium. In addition, tissue tropism is the bacterial reference for predilection for certain tissue for growth. Next is the specific adherence. Most bacteria can colonize a specific tissue or site because they can adhere to the tissue or site in a specific manner that involve complementary chemical interaction between two surfaces. Specific adherence involve biochemical interaction between bacterial surface component and host cell molecular receptor. The bacterial component that provide adhesion are molecular parts of the capsule, membrane, or cell walls. The receptor on human cell or tissues are usually glycoprotein molecule located on the host cell or tissue surface. Specific adherence involve complementary chemical interaction between the host cell or tissue surface and the bacterial surface. In the language of medical microbiologists, a bacterial adhesion attaches covalently to host receptor so that the bacterium docks itself on the host surface. The adhesion of the bacterial cells are chemical component of capsule. Cell wall, pili, or membrane, the host receptor are usually glycoproteins located on the cell membrane or tissue surface. And lastly, the biofilm formation. Some of the indigenous bacteria are able to contract biofilm on tissue surface, or they are able to colonize a biofilm belt by another bacterial species. Many biofilm are a mixture of microbes, although one member is responsible for maintaining the biofilm and may predominate. Now going back to the normal flora of the skin. Skin is constantly exposed to and is in contact with the environment. Skin is particularly adapt to a contain transient microorganism. Most bacteria on the skin are sequestered in sweat glands. The skin microbe found in the most superficial layer of the epidermis and the upper parts of the hair follicles are gram-positive cookie and corynebacterial such as Propionibacterium. These are generally non-pathogenic and considered to be commensal, although mutualistic and parasitic rules have been assigned to them. Staphylococcus aureus is found on the face and hands in individuals who are nasal carriers. This is because the face and hands are likely to become inoculated with the bacteria on the nasal membrane. The normal flora of the human conjunctiva. The conjunctiva is kept moist and healthy by the continuous secretion from the lacrimal glands. Blinking whites, the conjunctiva every few seconds mechanically washing away foreign objects, including bacteria. Lacrimal secretion also contains bactericidal substances including lysosome. This is little or no opportunity for organisms to colonize the conjunctiva without special mechanism to attach to the epithelial surface and some ability to withstand attack by lysosome. Pathogens which do not infect the conjunctiva are those to be able to specifically attach to the conjunctival epithelium. Newborn infant may be especially prone to bacterial attachment. Normal flora of Normal flora of the respiratory tract. A large number of bacterial species colonize the upper respiratory tract. The rarest are the always heavily colonized, predominantly with Staphylococcus epidermidis and Corynebacterial with Staphylococcus aureus, with being the main carrier site of this important pathogen. The healthy sinus, in contrast, are sterile. The pharynx throat is normally colonized by Streptococcus and various gram-negative cocci. The lower respiratory tract is virtually free of microorganisms, mainly because of the efficient cleansing action of the ciliated epithelium, which line the tract. Any bacteria reaching the lower respiratory tract are swept upward by the action of the conciliary blanket that lines the bronchi, to be removed subsequently by coughing, sneezing, and swallowing, and etc. Normal flora of the urogenital tract. Urine is normally sterile, and since the urinary tract is flushed with the urine every few hours, microorganisms have problems gaining access and becoming established. The flora of the interior utera, as indicated principally by urine cultures, suggests that the area by the inhabited by a relatively consistent normal flora consisting of Staphylococcus epidermidis, Enterococcus faecalis, and some alpha hemolytic streptococcus, some enteric bacteria and current bacteria, which are probably contaminant from the skin, vulva, or rectum, may occasionally be found at the interior utera. The normal flora of the oral cavity. The presence of nutrients, epithelial detritus, and secretion make the mouth a favorable habitat for great variety of bacteria. Oral bacteria include the Streptococcus, Lactobacilli, Staphylococcus, and Corynebacterial, with a great number of aerobes, especially bacteroids. The mount presents a succession of the different ecological situation with age, and this corresponds with changes in the composition of the normal flora. At birth, the oral cavity is composed solely of the soft tissue of the lips, cheeks, tongues, and palate, which are kept moist by the secretion of salivary glands. At birth, the oral cavity is sterile but rapidly become colonized from the environment, particularly from the mother in the first feeding. Streptococcus sulfation is dominant, and they make up 98% of the total oral flora until the appearance of the teeth 9 to 16 months in humans. The eruption of teeth during the first year leads to colonization. This remains another strain of Streptococcus ahead, strongly the gums and cheek of the teeth. Normal flora of gastroenteritis tract. The bacterial flora of the gastrointestinal tract of animals have been studied more extensively than that other site. The composition differs between the various animal species and within an animal species and human. There are differences in the composition of their flora which influenced by age, to with cultural condition and use antibiotics. The latter greatly perceives the composition of the internal flora.